Okay, can you see my screen?
Yes.
Yes, okay, excellent. Let's just go ahead from the beginning here, see if this is gonna work. So far, so good. Oh, I have my dog underneath my desk here, and I'll have to take away his squeaker toy for this talk. Totally forgot about that.
No worries, it's par for the course. On Zoom here.
I think everything appears to be working so far. You guys are getting the full run of the 35 minutes or so.
All right.
Yeah. Okay.
All right, looks great. So we'll probably start at like 3:02, 3:03, because usually it takes people a few minutes to trickle in.
Sure.
But yeah, I'll call on you when we're ready to start.
Okay, sounds great. Thanks.
Excellent. Yep.
Hi, Dr. Duffy. It's good to see you.
It's good to see you too, Liz. How are you doing?
Doing pretty well. I'm excited for the end of the semester. I'm ready for it to be over, but yeah, I'm doing well. How about you?
It's going well here. As long as we can keep those students healthy, it's all going to be good.
Yeah.
Looking forward to hearing your talk. I'm so glad you did this. Yay!
Yeah, me too. You know, when Dr. Cloth reached out, I was so excited, you know, and actually Nick Antonson messaged me and told me I should do it because he was going to do it earlier in the school year. So I'm so glad that I did.
Yes, we just keep taking advantage of our students.
Perfectly fair.
Although one of these days we will actually be able to bring them back to give talks for us. So that would be nice.
That would be even better.
I mean, Zoom's nice because you get to, you can hear from people all over the place. But, it'd be nice if you got to like leave your office, right?
So are you folks in the bio department still doing the 1-credit seminar course where students have to write a summary? Okay, good. I figured.
Oh yeah.
Good.
Looking to see who we have here that's in class. We've got Kelly, yeah, and Spencer, and Hannah, yeah, and Grace, yeah. There's a bunch of them who are here for that, but I'm sure they're here to hear your talk as well.
I'm actually here to just hear the talk because I got my seminars done.
Nice. Well, that's promising. Glad you're here, Kelly.
All right, we'll get started here in about 2 minutes. All right, we're gonna go ahead and get started here. And people will probably still trickle in. But I want to welcome all of you to the last biology seminar of the year. This is gonna be, the seminar is given by our alum from 2017, Liz Renner. But I'm gonna toss, before Liz gets started, I'm gonna toss it over to Amy or Dr. Lewis, who is gonna introduce Liz.
We're very pleased to have Liz back with us. As Dr. Close said, she graduated in 2017. She was a bio-Spanish double major, civitas, all kinds of great stuff, and ecology emphasis. So she definitely took all of our ecology classes here. She got so desperate that she had to drive up to SDSU a couple of times a week and take their fish courses up there because we didn't offer enough fish stuff for her. So that kind of explains what she's doing now. She had a couple of summer experiences here at Augie doing summer research with Augie professors. And then she got in 2016, she had an REU with the professor whose lab she's in right now doing her PhD. So it's great to get those sort of connections when you are just doing a little summer research, and then all of a sudden great opportunities come. So she's at Kansas State now in the Division of Biology, and she's here to talk to us about gizzard shad. Are gizzard shad bad? So we will learn about our gizzard shad.
All right, thanks for that introduction, Dr. Lewis. It's so good to see everybody. I'm so glad to be back with my alma mater. I have a lot of fond memories from my years in the Augie biology department, and a lot of late nights and early mornings spent in the Gilbert Science Complex and now the Froyland Science Center. And so, yeah, I'm just thrilled to be back with all of you. I'll go ahead and share my screen here. Okay. Can you folks see that all right? Awesome. Okay. All right. So as Dr. Lewis mentioned, I study gizzard shad for my dissertation. And so today I'll be sharing my talk entitled, Are Gizzard Shad Bad? Disentangling the Ecological Role of a Nuisance Fish in Small Impoundment Food Webs via Replicated Whole Lake Manipulations. And so just a brief outline of what I'm going to share with you today. First, Dr. Lewis did a great job of already touching on kind of my path from Augie to K-State. And I'll just briefly talk a little bit more about some of the research opportunities I had and the programs that I've been involved in for any interested undergraduates here that might be thinking of applying to graduate school. And then I'll get into a background on my study organism, the gizzard shad, and their ecology. And then I'll talk about the experimental design of my long-term study. And then we'll launch into my first dissertation chapter looking at associations between gizzard shad and limnological parameters. And then I'll get into my second chapter on fish community responses to shad introductions and removals, as well as looking at the importance of gizzard shad in predator diets in Kansas small impoundments. And then I'm going to wrap things up, bringing things bigger picture to talk about multiple stressors facing small impoundment ecosystems, and then we'll do a Q&A. But I thought I'd start with just a few fun photos from how I came to be a freshwater ecologist. So I grew up northwest of Crooks, South Dakota, on a farm between Crooks and Lyons. And I spent a lot of summers in my childhood at my grandparents' lake cabin on Brant Lake near Chester. And so from those early formative experiences of fishing off the dock and flipping over rocks looking for mayflies and dragonflies, I think that's where I really sparked an interest in aquatic ecosystems. And so that interest grew, and throughout middle school and high school, I got involved doing little science fair experiments in FFA, doing a variety of little tests on zooplankton and exposing Daphnia to herbicides and whatnot. And so that's my origin story. And so then when I was applying to schools, I was really drawn to Augustana because of the amazing undergraduate research opportunities there. And so my first 2 years at Augie, I bounced around to a lot of different labs and opportunities. And so I had the opportunity to work with Dr. Craig Spencer out on the Good Earth State Park at Blood Run, doing a stable isotope project with soil cores. And I worked in the conservation biology lab with Dr. Carrie Hall and Dr. Dan Howard, working with burying beetles, which is pretty fun. But then I took an aquatic ecology class, and we got to go do fieldwork out on Lake Alvin and on ponds around Sioux Falls. And I knew that was something that I wanted to pursue, and that was really my research interest. And so from there, I applied in the summer of 2015 for a job with the South Dakota Department of Transportation's Environmental Division. And so for that summer, I traveled around the state of South Dakota to all but one state. We didn't make it to Hardin County, but we made it to every other county around the state. And we did a lot of seining and surveying for endangered species at box culvert and bridge construction sites. And so that's where I got acquainted with some of the fish fauna of the streams of South Dakota. And I got excited about learning about stream sampling and monitoring. And so in that photo on the left there, that's me holding a federally endangered Topeka shiner from 9 Mile Creek in the James River tributary. And so from there, like Dr. Lewis mentioned, I decided to enroll in the Semester by the Bay program in Homer, Alaska in the fall of 2015. And while I was there, I got to take a lot of really cool coastal ecology courses, ichthyology classes, and I did a hands-on internship in a stream ecology lab at this nonprofit called Cook Inlet Keeper that does salmon stream monitoring. So they were evaluating the influence of climate change on macroinvertebrate communities and juvenile salmonids up in Alaska. And I had a blast, and I really enjoyed working up there, and that really influenced where I was going for graduate school, I guess. And then, I did the National Science Foundation has this research experiences for undergraduates program. And so that's what brought me to K-State in the summer of 2016. I got to design my own little experiment out on the Tallgrass Prairie National Preserve. And then in the middle of my senior year, my advisor for that REU, Dr. Gitto, actually contacted me and said, "Hey, Liz, I've got this really cool project in impoundments that I think you should apply for." And I ended up getting the assistantship and coming to K-State. And so here's a photo of me graduating in May of 2017. And then within 3 days, I had graduated, moved out of my dorm in Tuve Hall, and moved into a new apartment down here in Manhattan, Kansas. And I was on the water on a boat 3 days after leaving South Dakota. So it's been a whirlwind of an experience. But so I started my master's in May of 2017 on this 4-year project, and the opportunity arose for me to stay on and convert it to a PhD. And so that's how I'm working on a dissertation right now, which is pretty cool. There we go. And so you're probably wondering, what's the next step then? What do you want to do with your life, Liz, right? Like every graduate student or undergrad gets asked that at some point. And so my answer right now is that I'm actually thinking about moving away from academia. My passions really lie in applied fisheries ecology and management. And so right now my goal is to land a job as a fisheries ecologist with a state or federal agency like the Fish and Wildlife Service. And so this summer I'm really excited because I landed a paid internship with the Fish and Wildlife Service through this Directorate Fellows Program. And I'll be working on a long-term data set from Lake Mead, analyzing the impact of the quagga mussel invasion on these federally endangered Colorado pike minnow. So I'm really excited about that project. But for undergrads, if you're interested, rising juniors and seniors can apply to this program. And I can send you a link with more information afterwards. Okay, so now getting into some of my research, I wanted to just start by talking about dams. So dams are everywhere on the Great Plains. So there are hundreds of thousands of them across the Great Plains. And Kansas actually has the second highest number of dams in the nation. So you can see that map on the top. Each of those dots represents a dam. And most of these dams are small earthen dams on private land. So a lot of farm ponds that are fragmenting stream ecosystems across the landscape on the Great Plains. And so this has a lot of impacts on ecosystems, right? Because although dams provide societal benefits, so they provide opportunities for recreation and angling, they provide access to water for livestock, they're also causing fragmentation of ecosystems and changes in habitat across the landscape, and that has consequences for water quality. And so, yeah, small impoundments, you can see they're everywhere. And so now I want to talk just a little bit about my study organism. So I study the gizzard shad, Dorosoma cepedianum. And so they're a freshwater forage fish that are closely related to anchovies and herrings in the family Clupeidae. And so gizzard shad are really unique. They compete directly with juvenile bluegill and largemouth bass. And then they also can indirectly impact sport fish by reducing water quality. And so that top photo is a gizzard shad there. And once established in ecosystems, gizzard shad tend to dominate fish communities, and they resist management efforts for a number of reasons. They have a really unique life history, and so they're highly fecund, they grow really quickly, and they're also facultative detritivores, and that's really unique among fishes in that they can switch from feeding on plankton. When plankton resources are grazed down and low, they can switch to feeding on the detritus, the muck on the bottom of the lake, and they can metabolize that decaying organic matter. And so they're the only fish in these systems that can do that. And that's a pretty interesting ecological characteristic. And so it's known that gizzard shad can have some impacts on the limnology or the water quality parameters within small impoundment ecosystems. And so a number of studies have documented this relationship between high gizzard shad densities and higher rates of primary productivity, including high concentrations of total nitrogen and phosphorus, as well as chlorophyll A concentrations, which is a rough estimate of how much primary productivity is happening in an aquatic ecosystem. They're also known because of their foraging on zooplankton, they can depress the overall zooplankton biomass and size. So they're selective foragers, and they can prey on larger bodied zooplankton and shift entire communities in lakes towards having smaller bodied individuals. And so they do play a selective role in these systems. That relationship, due to direct competition for limited prey resources, can result in decreased sport fish abundance. And then their increases in primary productivity, they have been found to be associated with blue-green algae blooms. And so as you guys are probably aware, harmful algae blooms are becoming increasingly prevalent in reservoirs across the Great Plains. Here in Kansas, especially in large federal reservoirs like Milford Reservoir, almost every summer now we're seeing cyanobacterial blooms, and they're becoming increasing concerns with respect to livestock health and human health and just overall management of water quality in these systems. And so that's why we're interested in understanding the relationship between gizzard shad and these characteristics. And so just to kind of visualize what I'm talking about here. So here we have a gizzard shad feeding on a hypothetical lake bottom, right? So you see all this decaying organic matter. Well, when gizzard shad feed on that decaying organic matter, they then excrete nitrogen and phosphorus into the water column, right? And so it's thought that this nitrogen and phosphorus can then fuel algal growth and cyanobacterial growth within these systems. But what's less understood and what nobody has successfully quantified yet is whether or not the underlying limnological parameters in an ecosystem then directly influence the densities of gizzard shad in these systems, because it can kind of create this positive feedback loop, right? So the more gizzard shad are in a system, and the more they're grazing, the more nutrients they're releasing from the bottom, that fuels more primary productivity within the water column, and it's just kind of a spiral. And so we're interested in understanding that relationship and seeing under what conditions does it exist. And so here's a map of the range of the gizzard shad. So you can see the central part of their range is the Mississippi River Basin. However, in the past decade, the species has been undergoing range-wide expansion due to a number of reasons. And so these include illicit stockings, where we have what's called bait bucket anglers or bait bucket biologists who will go out while they're fishing for catfish or bass, and unwittingly they will dump shad that they collected from another water body in the morning into the reservoir where they were fishing then in the afternoon or evening, and they introduce shad into an impoundment where they weren't present. And then we're also seeing accidental introductions from waterfowl dispersing their eggs, as well as flooding. And so you guys probably remember there were some pretty extreme flooding events along the Mississippi and Missouri River basins in 2019, and that was the case here along the Kansas River as well. And that flooding created connectivity that allowed for gizzard shad movement into water bodies where they weren't previously found. And so because they are potentially a problem and they can become overabundant in these systems, fisheries managers are increasingly becoming interested in removing them via this low-dose rotenone manipulation method. And so that's what folks across the country want to know more about, whether or not it's effective for managing gizzard shad. And then I just wanted to touch on that South Dakota is really similar to Kansas in that both of these states are on the western edge of the species' historic range, and that shad management out in these novel habitats of these impounded streams that then turn into reservoirs can be more challenging because less is known about the ecology of these fish in this system. And so here is just to visualize a ridiculously oversimplified diagram of a small impoundment food web here in Kansas. And so what I want you to pay attention to is you can see gizzard shad there, the planktivorous fishes occupy this intermediate trophic level in these systems. And so they're acting, as I mentioned earlier, as benthic pelagic couplers that are feeding on that benthic detritus and resuspending those nutrients into the water column. And so since they're right in the middle of the action, you can see there's a lot of arrows going to and from that particular trophic link. They're really important to understand their ecology, and we don't know that much about it, so we wanted to quantify it and learn more. And so, yeah, just to summarize, gizzard shad are ubiquitous. They're still being intentionally stocked as forage and accidentally introduced into others. There's growing concern about them and their management costs, but we still don't really understand what they do. We lack a lot of basic life history information about how old they live to be, how fast they grow under different conditions and whatnot. And then no one has ever directly examined their effects on water quality using replicated lake manipulations. And so that's what we set out to do. And so my project is funded by the Kansas Department of Wildlife, Parks, and Tourism. And so several years ago, my collaborators, Ben Neely and Jeff Cook, set out to test this in a pilot impoundment. And so they did this pilot study in Montgomery County State Fishing Lake in 2011, and then they monitored through 2016. And they, in order to do this, they evaluated the relative abundances, the growth rates, and then the body conditions, so the relative plumpness of bluegill and bass before and after manipulation. And they found when they removed gizzard shad that the growth of largemouth bass and bluegill improved initially, but then it declined in subsequent years. And so they didn't understand why these effects were pretty ephemeral. And so they wanted to get a more mechanistic understanding of how gizzard shad were influencing lower trophic levels. And that's where my dissertation comes in. And so they approached my advisor, Dr. Gitto, to write a grant to study gizzard shad in more systems across the state of Kansas. And so the first goal of our project is just to examine the ecological role and collect some baseline natural history data in these systems. And then the second objective was to look at differences in water quality parameters across a range of shad densities to assess their impact on nutrients and primary productivity in these systems. And then finally, we wanted to look at their direct impacts on fish communities. And so looking at how predators' diets change across a gradient of shad densities, looking at the relative abundances, growth rates, and then just comparing the general structure and function of food webs between impoundments with and without shad. So here's a map of our 12 study impoundments around the state of Kansas. They range in surface area from about 17 acres to 330 acres. So when you think of small lakes or ponds, these are small reservoirs, so small impoundments. None of them are deeper than, some of them have holes that are pockets of 30 to 60 feet water, but the majority of them, the mean depth is around 5 to 12 feet. So they're very shallow. Most of them are eutrophic to hypereutrophic, so they have high concentrations of nutrients in the water column. They're very productive systems. And so our experimental design was actually really cool for this project. We tried to do a before-after control impact study. And so we had this ANOVA design with 3 impoundment treatment groups. So we had 12 lakes, and we were going to have 4 rotenone lakes with gizzard shad to start, 4 reference or control lakes with gizzard shad to start, and then 4 reference lakes without shad that we could use to compare. And we were going to study them over the course of 4 years. So 2 years prior to manipulation and 2 years after to compare changes. And then we also had a bit of, for my limnology questions, we had a bit of a regression design. So we wanted to look at relationships between shad densities and individual dependent variables across an environmental gradient. And so we were looking to examine these relationships between shad and things like, total nitrogen or total phosphorus concentrations using modeling and ordination techniques. And so before I get too far, you're probably wondering, what is this rotenone and how does it work? And so rotenone is a compound derived from the roots of Mexican yam plants. And this compound has actually been used in indigenous subsistence fisheries for centuries across Central and South America, which is really cool. And it works by blocking oxygen uptake at the gills. And so it effectively halts cellular respiration and leads to cell death. And because it can be toxic to aquatic organisms, you have to pass a course before you can get certified to be a rotenone applicator. And so here's just a visualization of the timeline for my dissertation project. And so we started sampling in 2017. 2018 in Kansas was a historic drought year. We had 2 unanticipated gizzard shad introductions in our control impoundments in 2018. And then at the end of 2018, in December of 2018, we applied our rotenone to our 4 treatment lakes. And then in 2019 and 2020, we did follow-up sampling to figure out what happened. And 2019 then was an extreme flooding year. And we did find that we temporarily reduced gizzard shad in 2 of our treatment lakes, and we eradicated gizzard shad from 2 of our other treatment lakes. And so here are just some photos from the gizzard shad removals with low-dose rotenone in December. And so you can see my collaborators mixing and applying chemical. We used siphon lines to pump it down beneath the water's surface so that it could be evenly distributed throughout the impoundment. And so for each impoundment, we had to calculate the water volume in the entire lake. And so we did that in little cross sections, basically estimating volume based on depth and the bathymetric characteristics of these systems. And then I went out in the days following and tried to quantify the mortality to see which fishes were succumbed to the gizzard shad and whether or not gizzard shad were effectively removed in these systems. And so the goal was to just track over time, then 2 years following rotenone manipulation, what happened to these food webs. And so before I really get rolling, I mentioned how we had an unanticipated gizzard shad introduction. And I just wanted to share that long-term field ecology studies are really tricky to control. And so we had a partial dam collapse at one of our lakes. We had historic flooding and droughts within a 20-month period. We had 2 shad introductions and a zebra mussel invasion in one of our control lakes right at the start of the study. And then, ecologists deal with equipment breaking and accidents happening all the time. And that just leads to missing data that makes it a little bit more difficult to run statistics, but we do the best that we can. And then unfortunately, there are a lot of gaps in Kansas in long-term water quality and fisheries monitoring. And I've heard that that's the case in some fisheries in South Dakota too. And so we are limited by the long-term data that are collected by agencies. And so the first step in this project was to quantify gizzard shad densities. So we had to determine how many gizzard shad are in these reservoirs. And gizzard shad are schooling fishes, and so they're pretty patchily distributed, and their densities are difficult to quantify accurately without the use of hydroacoustics. And so we used a combination of long-term standardized gill netting surveys and boat electrofishing to estimate gizzard shad densities in these systems. And so we were able to get this metric. Over here, you're seeing on the x-axis this shad CPE, that stands for catch per unit effort. So the number of shad that we caught electrofishing per hour, and that's our density metric. All right, so now getting into my first chapter of my dissertation, we wanted to look at those limnological parameters. So we wanted to just determine whether or not there were associations between shad densities and some of these characteristics. And we anticipated that we would see higher nitrogen and phosphorus concentrations with higher shad densities. We anticipated reduced water clarity with more shad, as well as depressed zooplankton densities in greater gizzard shad density lakes. And then specifically that we would see reduced densities of cladocerans and a increased dominance of rotifers and copepods, the smaller bodied zooplankton, in the presence of higher gizzard shad densities. And then we also wanted to look at the algal communities. And so we anticipated that we would see higher algal densities and in general, more cyanobacteria with gizzard shad densities, high gizzard shad densities. And so for this part of my dissertation, we went out in June, August, and October from 2017 through 2020 in a boat once a month, and visited 3 different stations per impoundment where we dropped anchor and we collected a number of water quality parameters. And so we measured secchi depth. You see on the upper left-hand corner, that's a secchi disc where we drop it into the water. And the depth at which you can no longer see it is a coarse estimate of the depth of the photic zone in which photosynthesis can occur in the top slice of an impoundment. We also collected water samples from the surface for nitrogen and phosphorus analysis. We did repeated zooplankton tows, both vertical and horizontal tows, to quantify zooplankton densities. And then we did pelagic algae grab samples and then fixed them in lugalls and settled them under an inverted microscope to count and quantify the community composition of the algae. And I filtered water for chlorophyll analysis and also used a handheld probe to conduct readings on conductivity, the pH, the total dissolved solids, so how much just suspended sediment and other particles were in the water, and then salinity. And we did vertical temperature and dissolved oxygen profiles. So we did just a whole smattering of limnological parameters that we were studying. And for all of these that you're going to see, I fit generalized linear mixed effects models to use regression to look at the relationships between shad density and each of these metrics. So we're going to start with total nitrogen concentrations. And so we found over the course of 4 years that total nitrogen tended to be associated with higher shad densities. And so we saw a significant fixed effect of shad density on total nitrogen concentrations, as well as a significant interaction between the shad density and then accounting for the effect of month in this system. And so we did see a pattern with nitrogen. We also saw a pattern with phosphorus. And so total phosphorus increased with increasing gizzard shad densities. And we saw a significant fixed effect of shad density as well as a significant interaction. And so both for total phosphorus and total nitrogen, that was supported in the literature and was in line with our predictions. Next up, looking at water clarity, we measured secchi depth and we saw, there we go. We saw that water quality marginally declined with increasing gizzard shad densities. And so we did see a little bit of an effect in which the more turbid a lake was, there tended to be more gizzard shad. And that's kind of a chicken or the egg question, which came first, the poor water quality or the gizzard shad. And so we're trying to get around that more in understanding that relationship. Next up, looking at total algal densities, we did not see a significant relationship between gizzard shad and total algal densities, which was surprising. However, actually, we did not see significant cyanobacterial density relationships either. So there were not more cyanobacteria in the presence of high gizzard shad densities. Next up, moving into the zooplankton communities. And so when I was counting zooplankton, we counted them and organized them into orders, right? And so the first order we're going to look at is the cladocerans. And so that includes the Daphnia that you've probably used in a variety of classroom experiments. And so we found that cladoceran densities declined with increasing shad densities, which was pretty interesting that we actually were able to detect a pattern with these shad densities. Next up, we saw a significant effect of gizzard shad on rotifer densities. And so this was also interesting in that we anticipated that we would see higher rotifer densities because there'd be that shift from grazing down all of the cladocerans to allow for rotifer dominance. And we did not see that. So gizzard shad were also, bringing down rotifer densities, which is really interesting. There we go. Okay, and we also saw a significant relationship with declining zooplankton densities in relation to increasing gizzard shad densities. And so that's consistent with our predictions, which was really interesting. So to summarize, we saw that higher gizzard shad densities tended to be associated with higher primary productivity metrics and nutrients, but there was no significant relationship between shad density and algal or sand bacterial densities. And so we didn't find evidence that gizzard shad were associated with harmful algae blooms. We did see that higher gizzard shad densities were associated with lower zooplankton densities, which could be a function of their foraging. And there were significant declines in cladocerans and rotifers. And so that begs the question, are they doing size selective foraging for larger bodied zooplankton? Next up, I'm going to move to my second dissertation chapter. So we're looking at fish community responses to shad introductions and removals. And I like to use the analogy of a Jenga tower. So if you think about a small impoundment food web as a Jenga tower, when you have an invasive species introduction, that is like putting a Jenga tower block into an already existing tower, right? And so we're interested in what happens to the tower if you put a block in, but we're also interested in seeing what happens to that tower when you pull a block out by applying rotenone to remove gizzard shad. And so we wanted to know whether or not littoral fish community composition, so the near shore juvenile fish community composition, changed following shad removals or introductions. And we also specifically, again, wanted to assess bluegill and bass densities. And we anticipated that we would see a greater change in response to shad introductions than to removals, just due to the impact of that initial invasion. And then we predicted that the response of bluegill densities would be more pronounced than those of largemouth bass. And so for this, concurrent with our limnological sampling on the same days that we were out collecting water samples, we'd also go out in the afternoon at these same impoundments, and we'd seine in 3 different stations per impoundment. So we'd go up lake near the inlet, mid lake, and down lake close to the dam. And so we would seine and then identify and measure all individuals down to the lowest possible taxon. And then we excluded the larval fish less than 15 millimeters total length for our analysis. And so again, I fit generalized linear regression models with the fixed effects of lake treatment group and status, so before or after a rotenone manipulation or introduction. And then I used what's called non-metric multidimensional scaling and permutational multivariate analysis of variance to evaluate change in community structure following shad removal or introduction. And so you can think of that as just a big multivariate ANOVA, but we're looking at relationships in communities rather than for a single organism. We're looking across many groups and multiple environmental variables. And then I also ran a kitchen sink model across all 12 seine fish communities over all 4 years, regardless of treatment, just looking at whether or not community structure changed in impoundments with and without gizzard shad. And so first off, with bass and bluegill densities, we didn't see a pronounced response in largemouth bass densities to shad introductions or to removals. But on the bottom left-hand corner, you'll see that we did see a weak but significant bluegill response in that their densities did increase when gizzard shad were removed, potentially due to a reduction in competition. Next up, we didn't see a difference in community structure in the impoundments following eradication. There was a lot of overlap in the community composition for these communities before and after gizzard shad. We also did not see within these two introduction impoundments significant differences in community structure after introduction, but we did notice that the differences tended to be driven by either the presence of gizzard shad, but then also the presence and abundance of golden shiner, which are a native analog to gizzard shad. They're a native forage fish, and so that was pretty cool to see. And then finally, for my kitchen sink model, did gizzard shad affect overall fish community structure? Yes, we found evidence that they did. And so across all of those groups, we did find some evidence that gizzard shad are influencing these fish communities. And so we didn't see a pronounced response in bass densities to introductions or removals. We did see a weak response in bluegill to removals, but not introductions. Community structure was affected by shad status. And then I just wanted to say that the potential signals of gizzard shad effects might be more difficult to detect in some of these impoundments, especially in small impoundments compared to large reservoirs. And that's for a variety of reasons. But small impoundments tend to be highly stochastic. So they're really dynamic systems. They change in water level fluctuations all the time. Because they're shallower, their mixing regimes tend to turn over a lot faster than larger reservoirs. And from year to year, they have wider changes in aquatic vegetation cover. And so all of those factors make it a little bit more difficult to detect patterns in limnology over time in these smaller impoundments. Okay, and then finally, my last chapter of my dissertation is looking at the largemouth bass diets in these Kansas small impoundments across gizzard shad density gradients. And so here are some photos of trophy bass from a variety of impoundments around the state. And so really managers around the state of Kansas just wanted to know what largemouth bass are eating. Are they eating aquatic invertebrates? Are they eating fish? Are they cannibalizing their own young? Or are they feeding on gizzard shad or crayfish? So they wanted to know if you removed gizzard shad from these systems, would that have a significant impact on their diets? And so we asked whether the frequency of occurrence of piscivory, so the consumption of fish in largemouth bass diets, was associated with gizzard shad densities. And we anticipated that we would see a greater frequency of occurrence of fish in largemouth bass diets in impoundments with higher densities of gizzard shad. We also wanted to see whether or not bass piscivory varied predictably with ontogenetic and seasonal shifts over the course of a summer in Kansas small impoundments. And so for this, we went out and we collected fish stomachs via daytime shoreline electrofishing. So these boats you're seeing are basically, there's a generator back by the operator, and that generator is hooked up to a switch where you can control the voltage and the frequency of pulse direct current that you're applying to the water. And that pulse direct current creates an electrical field. So those booms coming off the front are the anodes, so they're the negative charge, and then the bottom hull of the boat is the anode, or excuse me, the cathode, the positive charge. So you create that electrical field, and if a fish passes through that electrical field, they're temporarily stunned. It's called electrotetany. And during that time, their muscles can't expand or contract, so they're just temporarily frozen, and you can net them and then measure and weigh them and release them unharmed. And so that's a very common sampling technique among fisheries biologists. And so we also collected juveniles from those littoral habitats via the seining that I talked about earlier. And for each of these stomachs, we identified, measured, and categorized prey items as down as far as possible under a microscope, and then calculated the frequency of occurrence of prey items. And just to give you an idea of the scope of how many stomachs we've processed over the course of the past 4 years, I have not checked my database within the last week, but we're somewhere past 40,000 fish that we have measured, identified, and we've done diets. We've processed at least 22,000 of them now. And so we're sitting on a very large data set to allow for robust statistical analyses in these systems. And so here's a pooled table of all of the gut contents in largemouth bass from 2017 and 2018. And so you'll see that macroinvertebrates and fish were the two most dominant prey items, with crayfish and zooplankton coming up in third and fourth place. But we also found a variety of other organisms at times, as well as anthropogenic material. So we find not just hooks, but a lot of small plastic microfibers in the gut lining of these largemouth bass. And and plastic microfibers are becoming an increasingly prevalent ecological concern due to they're just spread across the landscape. They're everywhere. You can find them in the Arctic, you can find them at the bottom of the ocean. They're in every ecosystem, and we don't really understand their impact on organisms yet. And so we found that largemouth bass are utilizing gizzard shad as prey, but they're not preferentially foraging for them. And so in this little graph here, you see the vast majority of the fish that we identified were just pieces of scales and not enough vertebrae to make a positive identification. And so we were able to identify some as centrarchids or to genus level for the lipomus, the sunfishes. But we did find 26 positive identifications of gizzard shad in these systems. But that's less than even the rate of cannibalism that we were finding in largemouth bass diets. And so the vast majority of the prey fish that these bass were eating were actually bluegill and green sunfish. And so it didn't seem like they were preferentially feeding on gizzard shad. We also found that largemouth bass piscivory was not associated with shad density. So there was no significant relationship here between the frequency of occurrence of fish and the interannual mean gizzard shad densities. We also found that the relative plumpness of bass was not associated with gizzard shad densities. And so that was not a significant relationship. But we did find that piscivory tended to be highest at intermediate lengths and increased over the course of the growing season. And so they typically, juvenile bass would eat more zooplankton macrovertebrates. And then when they got to be 150 to 250 millimeters in total length, then they tended to switch to feeding on bluegill and some gizzard shad. But then when they got to be to trophy lengths above 250 millimeters, they predominantly tended to feed on crayfish instead of fish. And so that was a really interesting finding that I think changed a lot of attitudes among some of my collaborators, the biologists that manage these impoundments. They were not expecting that. And so my take-home messages across all of these chapters is that high shad densities are associated with lower total zooplankton densities, higher nutrient concentrations, and lower water clarity, but there was no relationship between shad and algal densities. We found some evidence that there are shad effects on fish communities, particularly on bluegill densities. And then we saw that gizzard shad are prey for largemouth bass, but they're not the most important prey resource. And that trophy bass in these impoundments are eating crayfish. And then overall, at high shad densities, gizzard shad appear to have these meaningful top-down effects on zooplankton communities through grazing. We also detected some evidence on fish communities of their bottom-up effects via direct competition for limited prey resources and then predation by largemouth bass and flathead catfish. And so their ecosystem effects, from what we've found, seem to be substantial enough to merit their monitoring and management attention. But they're not the most ecologically pressing concern in these systems. And so there are limited budgets for fisheries biologists, limited hours that you could be on the water, but they are worthy of at least counting and measuring their total lengths when they come up in nets each year to understand their dynamics over time in these systems. And so there's my argument that gizzard shad matter. I like using memes a lot in my presentations. And so I think that gizzard shad are playing a significant role in these ecosystems. And so the next steps, as I finish writing my dissertation, I'm set to defend in October, and I hope to graduate this December. And so as I'm finishing up my dissertation chapters, I'm also working on examining how land use in impoundments, their watersheds, influences water quality and shad dynamics. So the prevalence of row crop ag versus grasslands. We're looking at changes in sportfish growth via otolith aging. And so we collected a bunch of fish ear bones from the fish that we sacrificed for diet analysis. And you can age those ear bones like tree rings and calculate the distance between the tree rings on these otolith annuli to be able to figure out which years fish grew faster and which years growth was stunted. And then I'm also working to evaluate interactions between zebra mussels and shad in my study impoundment, using it as a case study since we had an invasion take place in the first year of this study. And I watched zebra mussels just blow up in this ecosystem. It was fascinating. And then I also collected thousands of macroinvertebrate samples that I hope to process at some point to look at changes between benthic and littoral invertebrate communities. And so to finish, I just wanted to talk about aquatic ecosystems. So they're getting walloped by multiple stressors. So a big one is anthropogenic climate change. And so lakes are warming across the Great Plains. We're seeing a lot of invasive species introductions, and these are having direct impacts on native biodiversity of fishes and aquatic invertebrates. We're seeing eutrophication and an increasing prevalence in harmful algae blooms, largely fueled by agricultural runoff and just the increased nutrient loading from agriculture as well as urban activity. And so we're seeing a lot of land use change. And then in general, a lot of these impoundments across the Great Plains were built from the 1940s to the 1960s and 70s. And so all reservoirs, after you build a dam, start filling with sediment over time. And that sediment acts as a nutrient trap. And so there's a typical aging process in reservoirs where over time, they become more and more of sediment traps where all of these nutrients are just bound in the bottom of these impoundments. And so it leads to reductions in sport fish productivity and water quality issues and the like. We're also seeing increased prevalence of pollution from chemicals to microplastics and more, and dams are creating habitat fragmentation. So endangered fishes like the Topeka shiner, so native minnows and other cyprinids are no longer have fish passage to create or to go about their spawning runs, and that's having significant impacts on native species. And so understanding what's happening in these systems can be a bit of an ecological dumpster fire, but it is our duty to continue to monitor them. And even in these novel ecosystems, these man-made lakes, they're still economically and culturally important, and we need to pay attention to their water quality and do a better job of managing them because they're not going away anytime soon. And so understanding gizzard shad ecology is just one way that we can facilitate better management of these systems and also conserve taxpayer and angler dollars in a time of unprecedented ecological change. And with that, I'd like to acknowledge my collaborators with the state of Kansas, as well as my dissertation committee, and all of the lab members past and present from the Gitto lab. It takes a small army to manipulate a food web and then study it over time. And so I'm grateful for all of the long hours that they put in helping me in the field. And then I'd like to acknowledge all of our funding sources and equipment sources, as well as the fact that my research was conducted on stolen lands. And with that, I would also like to thank all of my amazing professors and mentors within the Augie Biology Department who have supported me over the years and encouraged me to pursue a career as a freshwater ecologist. I couldn't be where I am today without your mentoring, and so I'm really grateful for your support. And with that, I'd be happy to take any questions.
As usual, let's go with student questions first.
I have a question. So I was kind of curious, did you, how is like the winter kill for the lakes, for lakes that have the shad in it?
Sure. So Kansas is a little bit different than South Dakota in that we have slightly milder winters here. And so we don't see as frequent of winter kills in these systems. We did document, there was at one point in the winter of 2017, there was in that January, a really cold snap for 2 weeks here in Kansas where it got below actually below zero, which is pretty rare here in the wintertime. And in the weeks after that, in the spring turnover, when the ice came off, we did see some winter kill, but it's not very prevalent here. Thanks for your question. If anyone has questions about graduate school or, something not necessarily related to gizzard shad, fire away. I'm happy to, so.
There are a few questions in the chat.
Oh, okay. Let's see here. Got to open this up. Let's see. Oh gosh, I'm going to stop sharing for a second here. There we go. Okay. All right. You stated that trophy fish switch over to crayfish. Is the reason for this known? Nutrition benefit, size increase? Yeah. Okay. Great question, Peyton. So, gizzard shad, we don't know what it is. And so that's never actually been quantified, but if I had to take a guess, I'm guessing that there'd probably be a higher protein content per gram of crayfish than in gizzard shad, and so that could be one role. But one thing that might be a mechanism here is that crayfish might be easier for bass to forage on. And so if you think about a mobile prey like a fish, it might be harder for bass to capture a fish, and they have to expend energy doing that in comparison to a crayfish, which can't move as quickly across the bottom of a lake. Good question. Okay, and then we have another one here. So how are gizzard shad decreasing the water quality if there's no significant increase in the algae? Good question. So we didn't detect a significant difference in our systems, but past studies have documented increases in algal densities. And so we're trying to understand, we were interested in seeing if that pattern held in these study impoundments. But in general, even though they didn't affect algal densities, they were associated with reduced water clarity, right? And so it's a little bit difficult to parse that out because in a lot of impoundments where we have high shad densities, we have the confounding variable of high common carp densities as well. And common carp are benthic foragers as well that, they carry out what's called bioturbation. And so when they feed on aquatic vegetation on the bottom of the lake, that ripping up and stirring can resuspend sediment. And I think that gizzard shad do a little bit of that as well. And so there are some impacts on water quality, but they weren't significant in these impoundments. Thanks, great question.
We'll open it up to professors too.
Dr. Matzner, I see your hand.
So these fish are native to the U.S., but they're kind of invasive in some areas or expanding their range or whatever. What do you think controlled them historically? Like, what kept them in check historically?
That's a good question. Well, historically, there weren't a bunch of dams and reservoirs across the landscape that created their optimal habitat. So it's thought that they evolved in the Mississippi River Basin's backwaters. So like slow, meandering side channels and oxbow lakes where there was, more still water and deeper water because they're pelagic spawners. So they spawn at the surface and they can engage in some surface feeding on zooplankton as well. And so that might be one mechanism, but perhaps just competition and predation by the larger bodied top predators in these systems could have played a role. We also have lost in the Mississippi River Basin the majority of the biomass of the historic grazers. So if you think about all of the sturgeon that have declined over the years and gars, a lot of them, there used to be, sturgeon that grew to be 5 or 6 feet long in some instances across the United States, and we just don't see that anymore. And so with that loss of biomass, there might also be shifts to community structure and ecosystem function. Did that answer your question?
Yes, thank you.
Okay, thanks. Sure, Dr. Goobles. Hi, good to see you.
Yeah, nice to see you too. Great presentation. So
Thanks.
exciting. I'm really curious about this idea that you can see how long a fish lived from the ear bones. Can you talk more about that? Like what that process is and what you're looking at when you're measuring? Is it a deposit of some kind of nutrient or what you're looking at there?
Sure, great question. So otoliths are really cool because they're these calcified structures in the ears of fish. And so these otoliths rattle around in there, and fish can use those vibrations to detect, it's connected to their lateral line system, and so they can detect sound and movement in the water around them by the rattling of those otoliths. And yeah, so they put down rings of calcium every wintertime as the water temperature drops, and you can use the spacing between those calcified rings to determine how much they grew, because a lot of studies have demonstrated across most taxa that there's pretty clean relationship between the distance between those rings and the body size, the total length and weight of a fish. And so it's cool to see in some of my study impoundments, we had bass that were 16 years old, white crappie that were 13 years old. And I just, I guess as an angler, you go out and you catch a fish and you don't really realize just how old some of these fish can be. But the survivors, they've seen a lot of things over time. So great question.
Yeah, thank you. That's really interesting.
Okay, so I have a question.
Sure.
So how do you get their stomach contents without killing them?
Sure. So we had to get permission to do sacrificing for all of these.
So all of those 40,000 stomach.
The 20-some thousand stomach.
Oh, okay.
Yeah, that we did sacrifice over time. So we had to calculate based on estimated, baseline populations of the different prey fish and sport fish in these impoundments, what we could take that was within the realm of natural mortality before we were significantly changing things with our sample collection too. And so, but we were able to use, long-term data and creel data from anglers to calculate that. So yeah, but there is a way to actually look at gut contents without killing fish, and it's called gastric lavage. And you take a little tube and you basically just push air into their stomachs, and then you have a bag to collect those stomach contents coming back out. So you forcibly puke the fish and then preserve those contents. It just takes a lot of time. Yeah. Sure.
So you just did it because of the time, or was it
in part for that reason?
More difficult to do with smaller fish, or what?
Yes, absolutely. Definitely difficult to do with smaller fish without killing them. And then also because we were interested in, this other question about growth rates, we had to sacrifice them anyway to get their ear bones. And so we tried to make use of as much of the fish as possible if we were sacrificing them. So I also took a bunch of tissue, like dorsal muscle tissue samples from just different representative taxa to try to do some stable isotope work. I don't know if that's actually going to get published, but so we tried to collect a bunch of different samples to look at different parameters. I collected nearly everything, it feels like, but we have all of these long-term data, and I will be publishing on it alongside my dissertation advisor for a while to come.
So you have a big freezer full of little bags.
I did. Fortunately, the freezer full has been reduced now to just a couple of shelves full over time. Yeah. Did any of the students have any questions about graduate school or programs like research opportunities? Sure, Elizabeth? Oh, sorry, I wasn't sure if you had your hand up.
So I'm considering going into a kind of similar career related to ecology, wildlife biology, and I don't know, just do you have any advice for that?
Sure, that's great. It's great to hear. I'd say the best piece of advice I could give to you right now in undergrad is to get as many research experiences and hands-on learning opportunities as possible. And so before you, hit the job market or are applying for graduate school, it really pays to get a variety of experiences and to try to get some of those internships and research opportunities to learn more about organisms, because hiring panels are looking for people with taxa specific skills or statistical skills, sampling skills, and so it's kind of a combination of what you know, who you know, and who has money at the time to bring you on as a student or to hire you. So yeah, just get as much experience as you can.
Okay, thanks. Good to know. I do have a couple of opportunities lined up.
That's wonderful.
Year.
I had a question for my Augie professors. How are you guys doing? How's the pandemic been at Augustana? So good to see you.
Really fun.
Yeah, I love pandemics. It's great.
Yeah, Yeah. So remind me again, were you face-to-face at all or like hybrid learning in the fall?
Face to face.
Face-to-face. Okay. Yeah. So we were
Well,
at
hybrid.
K-State.
We did some hybrid stuff too, but
Yeah. And did you have any outbreaks within the undergraduate population? Yeah.
Mhm.
Oh, so my partner and I got
after Halloween.
Oh, yeah, same actually. So in second week of November, my entire fisheries management lab got COVID. Like, and I got COVID. Yeah, everybody got COVID. So we distanced. I sanitized the classroom every day, and yeah. But it's a tricky little virus. It's good to see you, Dr. Olson Manning.
All right. Any last questions for Liz? Liz, you got any last questions for us?
No, I'm just so glad you folks invited me. Thanks for having me.
Yeah,
It was our
You did a
pleasure.
good job.
Thank you.
Yeah. This has been one good thing about COVID, that we've been able to have students come and give these talks virtually.
Yeah, for sure.
We wouldn't have really thought about doing that before, so good to see you.
Good to see you too.
Thanks for coming.
Sure. All right.
Right.
And then I think Dr. Cloth said that he was going to post a recording or something afterwards.
Yeah, is that okay with you?
Yeah, that's great.
Okay.
If I can just get the link to that, my advisor and like parents wanted to see this, because this is basically a dry run of my dissertation defense. So this was really great preparation for me. I'm so glad you folks invited me, because it forced me to put together this full talk. So
Excellent.
yeah.
Yes, we did record it, and I will send you the link, or I guess Dr. Close will, because it'll go to him.
Sure. Okay, great. Okay, well, it was good seeing everybody. Take care.
Yes, take care.
Thanks, Liz.
Good one. Bye-bye.